Bismillahirrahmanirrahim. This is a registration for the clinical laboratory methods for detection of antigens and antibody by Dr. Mona El Wassefy, former lecturer of Immunology Clinical Pathology Department, Mansoura University. The antibody detection is used to quantitatively and qualitatively evaluate normal and abnormal immune responses. Monoclonal antibodies has drastically and dramatically improved methods of antigen detection. Antigen detection is usually used to determine the presence of foreign proteins or compounds, for example, infectious agents or drugs. Here is the hepatoma, by which an antigen is introduced into a mouse, by which the mouse will produce B cell. The combining of both myeloma cell and B cell will enhance the formation of a huge amount of monoclonal antibody specific against this antigen, and this technique is known as hepatoma, which helped in the antibody and the antigen detection. The detection of antibodies and antigens depend on the formation of antibody and antigen complexes. One of the binding partner, I mean the antigen or the antibody, is defined, often labeled, and is used as a probe to search for the other partner. In its simplest form, a specific monoclonal antibody is used to look for a single antigenic epitope, and the formation of complexes monitored by the precipitation of the complex or by the presence of tag like fluorescent material or radioactive material or enzymatic material on the antibody. Reciprocally, use of model antigens to look for specific antibodies that recognize this protein determine the isotype like immunoglobulin G or M or E. When the antibody and antigens are present in equimolar ratios, equal amount, they form insoluble complexes that precipitate naturally. Light scattering or the formation of turbidity can be used to monitor accumulation of precipitate in the solution. Also, relative concentrations of the antigen and antibody are the most important determinant for the formation of the complexes. Maximum precipitations occur when the antibody and antigens concentration are equivalent in the zone of equivalence, and decreasing amounts of precipitation of severe complexes, of very small complexes, are formed in the zone of antigen or the antibody excess. In this, we will show a figure in a few slides later. Formation of these insoluble complexes can be used to quantitatively quantify the amount of antigens if a known concentration of antibody is used, and vice versa. The prozone phenomena here, when there is an antibody excess or antigen excess, and the suboptimal immune complexes are formed in this situation, in this phenomena can lead to a misinterpretation of test, and when large amounts of antibody are present, like in cases of multiple myeloma or polyclonal gammopathies, or when antigens are improperly diluted, like agglutination reaction. Here is the figure. This violet form is the zone of equivalence, and what's before it is an antibody excess, and what's after it is an antigen excess. We are measuring the concentration in the zone of equivalence, not before or after, because both of them are going to get us false results. The nature of antigen and antibody reactions, we must identify two terms: the term affinity, related to single antigenic determination, and the term avidity, which is the measure of the overall strength of binding of antigen with many antigenic determinants and the multivalent antibodies. Here is the affinity, single binding, and here is the overall attachment of antigen antibody, which is the avidity. We must also identify both terms. The specificity, we mean by this that the antibody combining site to react only with a single antigenic determinant. But if there is a cross-reactivity, so there are more than one antigenic determinant, or the ability of a population of antibody molecules to react more than one antigen. What are the tests used? Some of the tests are depending on the formation of immune complexes, like immune diffusion, nephelometry, turbidimetry, complement fixation. Other tests are depending on the antigen or antibody fixed to a solid surface, like agglutination test or ELISA test, or electrophoresis methods, like serum protein electrophoresis and immune electrophoresis, and immunofluorescent assay. What about immune diffusion? Immune diffusion is a simple technique by which antigen and antibodies are placed in a separate wells within a semi-solid support, like agar, then allowed to mix through the agar by diffusion. When a zone of equivalence is reached, a line of precipitation occurs, which is visible when the light is passed through the well. Also, there is a double diffusion. The first one is a single diffusion. There is a double diffusion agar, which is Octerlony analysis, characterized by relationship between different antigens. So you can use more than one antigen in the agar and test the antibody in the serum of a patient. What about the limitation of the single radial immune diffusion? It's an insensitive method, and by the requirement of a relative large amounts of precipitation, antigens, and antibodies. Also, it's time consuming, which is solved by placing the agar matrix in an electrical field, which drives the antigens and the antibodies together, and a technique called counter-current immunoelectrophoresis, which increases the efficiency of antigen and antibody complexes formation and often increases the sensitivity of the assay. Here is the single radial immune diffusion, by which the plate is impregnated with agar. This agar containing the antigen we are searching for, and those small circles are like wells by which the serum of the patient is going to be added. Then you are going to let this reaction for 24 hours or 48 hours according to the protocol you are using. This plate may be used for detection of immunoglobulins, all of them, and also may be used for complementary determination, but the long time needed for measuring and the subjectivity of measuring the circle may lead to false results. Here is the radial immune diffusions with its different pattern. Here the main difference is the presence of more than one antigen in the plate. Another test is the nephelometer, which also measuring the antigen antibody complexes. The formation of immune complexes in a solution is going to be measured by a spectrophotometer. The scattering of the incident light beam is used to detect complexes in dilute solutions of antigens and antibodies. There is a direct linear relationship between the antigen and the concentration and the optical density. Thus, the sample with high concentration of the antigen may require dilution for accurate measurements. The amount of optical density can be measured endpoint determination or also kinetic within rate nephelometer, which provides a more accurate quantitation of antigen levels. Here is the two terms, turbidimetry and nephelometry. If the detector is presented in the same line by which the sample is presented, this is a turbidimetry, but in cases of nephelometry, the detector is presented in an angle in relation to the presence of the sample. What about the test used for the antigen and the antibody reaction, like agglutination test or hemagglutination test? Here the antigen is a particulate by which the antigen is particulate. The reaction of an antibody with an antigen can be detected by agglutination or clumping of the antigen. When the antigen is an erythrocyte, so the term will be hemagglutination. The term agglutinin is used to describe the antibody that agglutinate particulate antigens. When the antigen is an erythrocyte, the term hemagglutinin is often used. All antibodies can theoretically agglutinate particulate antigens, but immunoglobulin M, due to its high valency, is particularly a good agglutinin. The test, for example, is the agglutination test, or by which it's a qualitative test. The antibody is going to be mixed with a particular antigen, particulate antigens, and a positive test is indicated by the agglutination of the particulate antigen. And the most common example here is the blood grouping, by which we are having three antibodies, anti-A and anti-B and an RH. For example, the first one here, the first slide, we can see any form of agglutination. The whole drop of blood is presented, so there is no antibodies against A and B, and no antibodies also against the D1, so this is an O negative. In the second one, there is an agglutination. Those clumps presented in the blue first spot, and here is the second spot for the B group is a whole blood, no agglutination occurs, but the RH here showing an agglutination reaction too. So this is an A positive, and in the second one here the B show agglutination, but the A didn't show any form of agglutination, so this is a B positive blood group. Also, there are quantitative agglutination tests. Those quantitative tests are going to depend on the process of serial dilution, which are going to be made of a sample to be tested for antibody, and then a fixed number of the red blood cells or bacteria, or often such particulate antigens is added. Then the maximum dilution, which is the titr, this is the results, are going to be reported as a reciprocal of the maximum dilution that gives visible agglutination, quantitative agglutination, hemagglutination test. Here is an example of the test in a serial dilution plate, by which you are going to see the maximum concentration in which agglutination occur. here is the agglutination at 1/64, so the titer is 64, and you must here notice there are positive and negative calibrator used here. Standard are used to ensure that the reaction is accurate. Here in the second one it's 1/8, so the titer is 8. Here is about 1/512, so it's 512 also, and vice versa. And the positive and the negative here, we mean by them there are standard to ensure the positivity of the reaction. And what is titer? Titer is the maximum concentration of the antigen. Maximum concentration, measuring of concentration. This plate is a common used plate for Salmonella typhi. This is the Vidal test used for exclusion of Salmonella infection, typhoid, we mean by this. In Vidal test, we know that we're having the somatic antigen, the O antigen, and flagellar antigen, the H antigen. Fourfold rise in antibodies titer over time when paired dilution of serum samples are tested with any of these antigens, while more specific tests are now available. This test is still considered useful in monitoring typhoid fever in developing countries, and this test is used for the RF measuring also. It contains a standard to ensure the positive and negative control to ensure the positivity of the test, and also it contains the reagent, and we are going to see an area of agglutination same to the ABO and same to Vidal test. What about the indirect agglutination or the passive agglutination? Here the antigen is not normally found on the cell surface. For example, erythrocyte, latex, gelatin, or silica are used for this purpose. And for example, rheumatoid factor, measuring the antibodies to Streptococcus A, and measuring the antibodies to Trichinella spiralis, and the antibodies to Treponema pallidum, and HIV antibodies also. What about the reverse passive agglutination? In this situation, we can see that the antibody, rather than the antigen, is attached to a carrier particle. The antibody must still be reactive and is joined in such a manner that the active sites are facing outward, so it could bind to the antigen and called an agglutination reaction. Here the antibody is presented over the slide, fixed, and the antigen added to the antibody, forming an agglutination over this slide. What are the uses? It's used for rapid identification of group B Streptococcus, Staphylococcus aureus, Neisseria meningitidis, Haemophilus influenza, Vibrio cholera, and others. What about the agglutination inhibition? Here the positivity is in inhibition of the agglutination reaction, by which in these situations we're having an antibody and antigen, so there is a competition will be formed between the bound antibody and the particulate antigens, the particulate and the soluble antigens for limited antibody combining sites. And if there is no agglutinations, this is indicators of the positive reaction. For example, used in some sort of detection of drugs like cocaine and heroin. What about the hemagglutination test? Hemagglutination test, here the reaction is used same principle, except red blood cells are the indicator particle. In this type of test have been used to detect antibodies for certain viruses like rubella and mumps and measles and influenza, hepatitis B virus, herpesvirus, respiratory syncytial virus, and adenoviruses. Here is the virus like orthomyxovirus or paramyxovirus are having some sort of tropism toward the RBCs. So these viruses are going to be, if they are incubated with the person RBCs, they are going to bind to those RBCs. Then if the antibodies against this virus is presented in the serum of the person, you are testing his sample. So the virus will bind to its antibody and will leave the RBCs. So there is agglutination inhibition reaction for the positive test. What about agglutination mediated, antiglobin mediated agglutination, which is the comp test? We are having two forms of comp test, the direct and indirect. In cases of direct comp, we are using the patient RBCs, searching for the presence of antibodies over the patient RBCs, allowing the hemolytic reaction to occur by adding anti-human globin. If it could bind to the antibodies over the surface of RBCs and form agglutination, so this is a positive comp test. And the indirect one, here we are having a blood group O containing the D, O positive, so it contains the D. If the person is having anti-D, it will bind to the RBCs with O positive and will form an agglutination reaction too. That's all what we want to know. Thank you for your attention. Best of luck.